Good afternoon, everyone. Welcome to the 11th ALA webinar, March 2023. Really excited to be here. Thanks for joining us. Before we go any further and I introduce our glorious speakers we've got today, I'd like to acknowledge that we're broadcasting today from the CSIRO Black Mountain campus, which is in Canberra. And of course, that's on Ngunnawal and Ngambri country. And I'd like to pay my respect and respects of this organisation to the elders, past and present, of those people. And those words, past and present, I think are particularly relevant today to our topic of conversation, where we're discussing advances in genomics, and particularly as they relate to the biological collections. Now that I'd say past and present is a relevant concept there. Obviously, genomes are the DNA, right? The store of millions of years worth of evolution. So there's a long time scale that we're discussing here. But then the specific genomes that we're going to be talking about and our speakers today are expert in are those from biological collections that have been built up perhaps over decades or centuries. And I think it's really interesting to consider that on top of that long history, we now have new methods that are casting light on biological diversity and its origins and its patterns that are advancing every day. And so you have this very long legacy and yet also this very immediate set of change and knowledge that we're coming to learn from. And so I'm really excited to hear more today from our speakers about that topic. We have 4 speakers today, just to make our life a little bit more complicated than usual. First of whom is Jessica Fenker, who's calling today from Melbourne Museum. Give us a wave, Jess. And we have Erin Hahn, who's also here in Canberra at CSIRO. And we have Kiva Connolly and Catherine Hall, who are from the Australian Reference Genome Atlas. So I'm really excited to hear what you guys have to tell us about today, about a topic that I am fascinated in, but in no way expert in. So very excited to hear what you've got to say. Our first speaker today is Jess. Jess, would you mind sharing your screen? Lovely. Thank you so much. That's fine. Over to you.
Hi, everyone. First, I want to thank you for inviting me to present this talk today. And I want to acknowledge the Woiwurrung and Boonwurrung people of the East Kulin Nations, which lands I am presenting and living today. So firstly, I will introduce myself. So my name is Jessica Fenker. I'm originally from Brazil, more specific to the south of Brazil, Paraná state. And I moved a few years back to Australia to do my PhD at the Australian National University. So during this time, I investigated the principal factors that shape the adaptation and diversification of lizards and snakes in savanna environments, both in Brazil and Australia, from a macroecological view, kind of a comparison between them, but also at the population and landscape genomic studies. Although these species and the stories of these environments are independent of each other, and the structure of these environments is quite similar, which makes it a really exciting comparison. After that, I completed a research fellow at the University of Michigan, but because of COVID, I did it entirely remote from Australia. And then I worked as a data analyst at the Atlas of Living Australia in partnership with Epigenomics for a while. Now I am part of the Museums Victoria. I think I joined a couple of months ago, working in an ARC research grant. But before I jump into my research and talk more about it, I thought it would be interesting to highlight a few points to provide background to this conversation. So for more than 90 years, museum collections have been used for experimental biology. The traditional roles of collection include taxonomic studies, as displayed here with the school of this kangaroo, but also as public educations, as like in museum stalls. Increasingly, these collections moved from not only telling species stories, but also to be a source of new information and cutting-edge research, in a way that the main role of collections has been greatly expanded. The use of molecular biology in natural history collections began around the 70s, when some researchers moved small pieces of tissues from DNA extraction for specimens they collected and deposited them in the museums. Now, these tissue collections are used for a wide range of objectives, going from conservation to studies of infectious disease to even evolution of specific traits. So why are natural history collections, and more specifically tissue collections, important? Well, museum collections can provide a snapshot of the biodiversity and a reference point for a given species at a given point in space and time. So tissue collections provide the necessary material for DNA and RNA extractions, and especially relevant if it covers a vast time span. So there are fossils from millions of years ago, like this reptile vertebrae here from the Quaternary period, through to specimens collected today. But also, within an extant species, there can be multiple specimens spanning hundreds of years in a collection. So with this, you could not only have access to such data without the museum's collections. The metadata associated with the voices and tissue samples is also quite relevant, especially in studies that you aim an integrative approach. So if you have the genetic material, call records, coloration patterns, and the distribution information of a species, for example, that is deposited in a museum, you can relate all this data and generate more precise results. And finally, collections are also invaluable for data storage. So initiatives such as the OzMemo and the Australian Amphibian and Reptile Genomics that are aimed to sequence the genome of the Australian diversity for diverse purposes, including conservation, they are only possible because of this material hosted in museums. Environmental DNA, which is a method where researchers collect a sample for water or soil, for example, aimed to identify all the species that occur in an area, need the genetic material from a diverse range of museum samples to compare them with the material they obtain. Now, let's jump into some examples of how these museum collections can be used for research, using some research that I did. So most of the work I do is focused on lizards and snakes as organisms of study, and there is a reason for that. So you can find them in different types of environments, from the top of mountains to deserts, and they are ectotherms, which mean that the temperature of the body is related to the temperature of the environment, and they also have short generation time and relatively limited mobility, which make them really excellent organisms of studies, ecology, and evolution. So I conducted a phylogenetic study on the telegeckos of the Australian muscinotropics to determine the relationship between different populations. This one is an example of one of these species. And this used to be the known distribution of Geharia australis, one of the main species that I worked with. After getting tissue samples from museum specimens spanning the species' full distribution, I performed population genomic analysis and discovered that the populations were actually different species with no gene flux between them. So if you look at this principal coordinate analysis here, you see different clusters of populations. If this was the same species, you would expect a more continuous pattern instead of these clusters. And if you also take a look at this fast structure here, you will see that there's no of this yellow or of this green, of this red in the blue, which means that there is not many gene flux between this. So this was supported by the morphological evaluation of museum samples, where we found sufficient evidence to distinguish and describe 3 new species. Here's another example. For a long time, children and Stinson's python were considered distinct species based on their color pattern. The thing is that it's really tricky to distinguish these species by the color because they are highly variable, so the researchers would just rely on the distribution to distinguish them. Everything that was in the north, in the top end region, would be Children's python, and in the arid or in the south region would be Stinson's. However, in a study that I collaborated in phylogeography and systematics of the world's smallest pythons, we discovered that they are actually the same species. So if you look at this principal coordinate analysis again, you see that they are overlapping, both children at the same area. This diagnostic was supported by different methods, from genomic data to head shape and body form. I brought these 2 examples because if you think that a species are the unit that you use in conservation initiatives, this kind of studies that redefine a species has really important conservation and management implications, when a species that you think would have a broad distribution would have actually a really restricted one, and vice versa. This brings me now to the current project that I've been working on, the morphological and ecological shifts in Australian reptile communities assemblages over time. So this project is part of an ARC grant, as I mentioned before, led by Jane Melville at Museums Victoria, with many partners across Australia and abroad. Just to provide a bit of background before jumping into the aims, the relationships between organisms and the environments across large spatial scales, in this case here, entire Australia, provide significant insights into the processes that shape modern biodiversity. However, macroecological insights from current environment conditions have limitations because they fail to account for historical events. For example, without any knowledge of the paleoecological perspective, we would have little understanding about how the past extinction of Australian mammalian megafauna has shaped the current marsupial communities. Yet, such work is almost completely lacking in the Australian reptiles, despite its being of global importance and the presence of megafauna equivalents in lizards and snakes. This is especially relevant as Australia has more reptile species than any other country on Earth, with a high endemic diversity. By basing our research on the Eastern Australian reptiles, we take advantage of a unique combination of a really high diversity and comprehensive phylogenetic framework, a strong climatic gradient, and detailed quaternary climate data, and also appropriate fossil sites. With that, the main aim of our project is to quantify how reptile community composition, both in terms of taxonomy or the different species, as in size and shape diversity of the body form, has changed over the past 500,000 years. And this will be based on data between and within our fossil sites and comparing fossils and modern reptile community assemblages. And second, we want to determine how these final changes correlate with climate and vegetation shifts in the past and present, but also in the future, using a compilation of database of reptile communities through time with detailed environment and habitat types. So I mentioned before about using fossils to answer our questions. These fossils are mainly from cave deposits across our area of interest. So we have since caves deposits from the Chalago Caves in Queensland to the Narracud Caves in Southern Australia. This is actually the first study to utilize a huge amount of reptile fossil data already stored at fossil collections, and especially at Melbourne Museum. And me, being relatively new to the paleontology world, need to say that I was impressed by the amount of fossil that is available and also about how tiny they are. If you imagine a vertebrate of a sun skink that you saw walking in your garden a few days ago, you can imagine about how tiny I'm talking about. So the strategy we will use involves three methodologies. From the fossil obtained in these caves, we will use 3D scanning to verify changes in the species body forms, and I will explain a bit about it a bit more in a few minutes. We also aim to extract DNA from these fossils, which will reveal information about the evolutionary history, and combining the information about the reptile communities obtained by these methods and the location of the caves, we use ecological modeling techniques to understand the changes in climate conditions over time. So let's talk first about CT scanning. I will just mention really briefly about these methods. So CT scanning is a non-invasive technique to see the fossil inside out. It's quite similar to the 3D scan that we do in humans. So here is an example of a machine that we can use, and we will posit here the specimen or the fossil, and these over projections over 300 degrees and using reconstruction algorithms will generate 3D visualization and cross-sectional images. In this work, we are developing new techniques to scan fossils in bulk, which will allow us to scan multiple fossils at the same time, while continuing with a really high resolution and image quality. We can do a lot of things with the data obtained from CT scans, as measure and compare the size and shape of distinct reptile structures, but also to observe changes through time when we compare the CT scans of the fossils with recent specimens. Now, let me talk about the ancient DNA component of the project. So how is ancient DNA compared to current DNA? It may sound super fancy, but the only difference is that ancient DNA comes from very old samples. It's pretty cool that we have now the technology to take DNA out of these old specimens. But nothing like in Jurassic Park, though, where they used the ancient DNA to extract DNA from insects in number to bring dinosaurs back from extinction. However, extracted DNA from old material can be quite challenging. In most cases, the quality of this old DNA is very poor and degraded and fragmented. And also, work on ancient DNA is quite different to work in DNA from present day. It requires a special environment with limited access of people and regular cleaning with bleach to avoid contamination. As you can see in the image here, a complete personal protective equipment is essential. This is because the last thing that you want is to sequence your own DNA or risk any type of contamination when working with the samples. In our study specifically, we will be using ancestral DNA to help answer questions about which species there are from and whether the reptile diversity has changed over time and space. And finally, which it brings me to the last method, the ecological modeling. And here is just to highlight one more time that we have selected fossil sites across the East region, which branch the time span of the origin of current reptile diversity from this region. And also we'll be able to access how the climate shifts influence the current herpetofauna community. So if you pay attention at this graph here, you will notice that over the years, there was an oscillation in the world temperature condition, and it was in a cyclicity form between cold and dry and warm and humid climates. So you can imagine that the species that would survive in a cold and dry environment, this is just an hypothetical example, would not be able to survive in a hot and humid environment, or they will need to change, they will need to adapt, or they will get extinct. So to understand how these changes affected the species and to generate the models, we relate climatic and environmental variables that are important for our reptile distributions, and combining them with the species distribution that we have and using different species distribution model algorithms, we will be able to predict the presence of the species in the present and the past, and also the estimations for the future. And this is a work still under construction, and the only thing that I can tell you now is that we are happy to know that we have some viable DNA from marsupials that we extracted from the same caves in same regions. And we did, looking at the fossils, know that the morphology of the fossils, know that we have species that no longer occur in this region, probably related to the climate shifts that happened in the past. And that's it. Thank you all for today. And if you have any questions, please let me know.
That's wonderful. Thank you, Jess. And for those who do have questions for Jess, you may have noticed in your browser that there is an option to add questions, and we will be have time for questions at the end. So please put those in. There's also a poll if you want to tell us some information about yourselves as the audience. Erin, I believe you're our next speaker, if you wouldn't mind sharing your content. And yeah, over to you. Thank you.
That's good. Coming through okay?
Yep, looks great. Can hear you fine. All good.
Great. Thanks for checking. Thank you, everyone, for having me today. As Martin said, I'm based in Canberra, and so I'd like to acknowledge that I'm seated on Ngunnawal country and pay my respects to elders past, present, and emerging, and extend that respect to the traditional custodians of the lands upon every which anyone joining us today is seated. He also mentioned that we're going to be talking about time. So I'm going to be telling a little story about how we're turning back the clocks and using a bit of a time machine that we've created at the Australian National Wildlife Collection to understand how species are changing in response to climate change. I'm seated within the National Research Collections of Australia at CSIRO, and this together comprises 7 different collections, which each specialize in their own taxonomic group, but together we house over 15 million specimens. Together, all of these specimens represent different time points, little snapshots of how species are responding to all sorts of various threats over the last 150 years or so. And this might be threats with relation to climate change or novel pathogens or introduced species. And as we can see on this graph on the right from the ALA, the number of specimens that we have in our collections is increasing. And so this is giving us a lot of power to look back in time and see how species are responding. And as Jessica mentioned, we're starting to apply all sorts of fancy high-tech techniques in collections in relation to genomics in order to learn more about how species are responding. There's a variety of different tools and applications that we use and apply to museum specimens, one of which, as Jessica mentioned, is generating reference sequences to be used in studies like eDNA to understand the species composition of our environments. Another is that we provide sources of material to assemble reference genomes that can be used by researchers all around the country and the world in order to study species. Also, because we collect specimens, not just individuals representing a species, but lots of different specimens across ecosystems and through time, we can use those sequences to understand how individuals within a species or in a population are related to one another and to other species in their ecosystem. Another layer that we're starting to get from museum specimens is that of the epigenome. So you're familiar with the genome is the A's, T's, C's, and G's. The epigenome is all of the mechanisms that control the genome and express what it does. These come in a variety of different flavors. Perhaps you're familiar with DNA methylation, so the addition of a little methyl group on the DNA. It also comes in flavors like different chemical modifications to the packaging of the DNA into histones, nucleosomes, and to the greater chromosomes. All of this information is additionally important because, unlike the genome, the epigenome gives us information of how individuals are responding to their particular threats of their day. The genome evolves over long periods of time, but the epigenome can change within the span of an individual's lifetime. So by looking at the individual's epigenome signatures, we can understand how they've changed in response to a whole bunch of different environmental parameters. At the Australian National Wildlife Collection, we have specimens that are preserved in a whole bunch of different ways, either in spirit, in liquid, or in dry in different forms as hide or bones. And more recently, we started collecting fresh tissue samples. And as Jessica said, these are really, really important to serve as genomic resources. We can get DNA out of pretty much any of these specimen types, but it's those fresh tissue samples that give us the most reliable sources of DNA. But that's when you're looking at genomics. For epigenomics, it gets a bit trickier. We can, of course, go and sample those fresh tissue reserves because they're essentially the kind of tissue that you would get in a modern laboratory, and you can throw any assay that you want at it. Unfortunately, we've only started sampling these fresh tissues over the last 20 or 30 years or so. So if you want to get information about how species were responding prior to that, we have to look for other sources. We could try looking in the bone vaults or looking at hides, but these dried tissues are not preserved with their internal organs intact. And so if you're looking for how a species is responding to, say, a novel respiratory virus, the bone or the hide isn't necessarily going to tell you that information because the epigenetic response is tissue specific. To get tissue-specific information, we have to look for specimens that are preserved whole, and that really only exists in our spirit collections. So these are specimens that are sometimes preserved with formalin or not, and then stuck in ethanol for long periods of time. They're wonderful resources for morphological characteristics because the whole specimen is preserved. However, for a long time, they've been thought to be completely devoid of DNA because of the chemicals that are used to preserve them. So formaldehyde is used to lock everything in place, shut everything down so that you can examine these specimens for decades to come. Formalin also has the nasty habit of when you try to extract DNA from these types of specimens, it comes out heavily, heavily fragmented, even more so than that ancient DNA that Jessica was talking about. And so for the last 20 or 30 years or so that we've been doing genomic work in collections, most people have ignored the formalin vaults. My team, as of last year, and a few other groups, have been developing methods to finally crack open the spirit vault and get out DNA from these sorts of specimens. You don't have to pay attention to the details here. Just know that now we have a system in place where we can vet individual specimens for their suitability for sequencing, prepare the tissue and extract DNA, do some sequencing and specialized bioinformatics to now reconstruct whole genomes from formalin-preserved museum specimens. And with that method that we published in Molecular Ecology Resources last year, we've since gone even deeper on some specimens, and we've recovered a whole genome from a 1905 eastern water dragon and even teeny tiny specimens of tuna larvae. And we're starting to get the same kind of coverage that we're able to get from fresh tissues. So we're pretty confident that we fixed formalin. It's not the absolute kiss of death that everyone thought it was. And this is really exciting for researchers who study reptiles, amphibians, and fish, because these specimens are almost exclusively preserved in formalin, and now we have a way to do genetic analysis. With the confidence that we can get DNA out of formalin-preserved specimens, we're now also opening the door to doing museum epigenomics in these tissues. Because if you're preserving the DNA, you're likely preserving all of those epigenetic marks that are sitting on top of the DNA that can give you that environmental response data. And in fact, anyone working in epigenetics in a lab with model systems knows that you often use formaldehyde in order to lock in place those chemical modifications for later study of how the epigenome is structured. So my group decided to take 2 of these assays and see if we could get them to work in museum collections to take advantage of that formalin rather than seeing it as an impediment. Of course, we didn't want to start going into the vaults and using precious museum specimens. We needed to create an experimental data set in order to start practicing our methods. And to do this, we created mock archival specimen sets with laboratory mice because they have a really nicely characterized genome. We can take fresh tissue from them, which we can do control assays with the fresh tissue, and then we can put them through our processing just like we would with a normal museum curated specimen, and then see how our assays do. So about 6 years ago, we started down this path, and we optimized these assays very, very heavily for use in archival tissues. It involves many, many more steps than for genome sequencing. Suffice it to say, we now have a method where we can go in and reliably get epigenetic information out of museum preserved specimens. But the key selling point is whether or not it looks like that information that we get from fresh tissues. And what we see is on the left-hand side, these are those same assays that pull out epigenetic information, but applied to fresh tissues. And what we see is this sort of funnel plot, which tells us about activity of genes across the genome. So this is giving us information about which genes are on and which genes are off. And both of these assays applied to fresh tissue give us this nice little funnel. When we do the same thing and apply it to archival specimens using our heavily optimized methods, we see a similar signal. So now we're seeing that we can pull out information from these archival specimens that can then be used in conjunction with comparison to fresh tissues to give us environmental response information. Now that we've got it working in our mock archival system, of course we want to go and test it in real museum specimens. And to do this component, we teamed up with researchers at the University of Queensland, Celine Farrar and Nicola Jackson. They study eastern water dragons and their response to urbanization in Brisbane. Celine and Nicola have this really wonderful modern data set that comprises behavioral morphological data, their response to a fungal pathogen that's been introduced, and they've started to map out how these species is genetically differentiated across the range of urbanization in Brisbane. But they really wanted to compare the species to what it looked like before Brisbane was heavily urbanized. And the only way to do that is to go into museums. And they, of course, thought that formalin was going to inhibit their ability to do so. But they teamed up with us, and we sent Nicola into the collection at the Queensland Museum to find specimens that would be suitable for sequencing and have sufficient tissue to also process them with our archival assays. Just cutting to the chase, we're able to deliver to them genomes that were sequenced to levels that we can then call SNPs and give them that genetic differentiation information from old samples compared to their present day samples. And when we apply our method for extracting archival epigenetic information to the samples, we see that same funnel plot. even in specimens that were as old as 1905. And so we see that the age of the specimen doesn't really matter in terms of our ability to pull out epigenetic information, which now gives us a lot more confidence to go back into the vaults and start analyzing these specimens in a new way. So this work was the brainchild of my team leader, Claire Holloway. She assembled this wonderful team across CSIRO, and we've all been working diligently to make this dream a reality. And of course, we have our collaborators in the ALA and at UQ, and this work was supported by the Environomics Future Science Platform, Bioplatforms Australia, and also AGRF. Thank you.
Thanks, Erin. I mean, this isn't my field, I should point out. So I'm a field ecologist originally by training, and it's been, like, those two talks already have been fascinating and revealed some things that I'd love to ask you more about. And in fact, that lots of people already started queuing up their questions for. I won't slow us down now, but I just want to prep you that people are fascinated by the research that you've been describing. Now, Kiva and Catherine, you're sharing the final talk. Which of you is going to start first? Oh, Catherine, you're way ahead of me. Lovely. I can see your browser. Yep. I can't hear you, though. I think you're muted.
Can you hear me now?
I can. That's wonderful. Lovely. Yep, all good to go. Thanks for that. Over to you.
Oh, thanks so much. And thanks so much for inviting us to talk today. Kiva and I are coming in further down the road than Jessica and Erin. So what we're doing is very exciting, and we're very grateful to be involved with this project. As more and more people are generating more and more sequence data, we're finding that people are finding other material to compare what they're making to. And so out of that need was born the Australian Reference Genome Atlas project. And our main ambition or mission with this project is to help researchers just begin to find, discover and interact with genomics data in whatever format they are. And as they are spread across a number of resources globally and domestically around the internet and in other private repositories. So I'll just explain a little bit about the way that the project is being structured. I'm working with the Atlas of Living Australia, but Kiva, who will be talking in a little while, is with the Australian Biocommons. And these two organisations, together with Bioplatforms Australia, have got together to realise the vision of creating this reference genome atlas. And we also have some additional funding from the ARDC to help us look at particular use cases for different sets of genomics data. We are an NCRIS platform when we launch, and we're very grateful to be a service to researchers around the country. So we've heard a little bit about why genomics is very exciting. It's fantastic for looking back in time, for being a bit of an archaeologist in terms of furrowing down a phylogeny to see why species have come into being in our current time and space, and the processes that they went through in terms of their evolution. We can observe a lot of that through the phylogeny. But it's also increasingly important nowadays with environmental threats and climate change and things like that to understand what's happening with genomes in our current environment. And the government has actually recognised how important genomics study is in terms of our prosperity and our success in the future in terms of our health and medical arenas. For instance, it's not just things like coronavirus, it's also aspects like vectors for illnesses and diseases, for things like arboviruses that come from mosquitoes. It's very important. It's also very important to our agriculture industry as we begin to develop species that are able to withstand climate change. They may be more salt tolerant. They may need to be able to withstand bushfire or smoke, have smoke tolerances and things like that. So in terms of going forward for the future for Australia, the government has identified that this understanding genomics and doing a lot of research in this space is incredibly important. And probably so that they end up crafting policy that is not just reactive, that it's actually proactive, and we can, in understanding these variations and impacts of conditions on genomes, we can actually tailor policy to suit the conditions as they are. Around Australia, the Biocommons has recognised that there are probably about 15,000 life scientists who are working in some way with genomics data. So there's a very great need for these researchers to have quick and easy access to all of the genomics resources that they need so that they can spend less time searching and more time answering questions as more and more data comes online. The problem with it is, though, that genomics data are variable in format. We've already seen some examples of the types of things that there are. There's barcodes, there's whole genomes, there's mitogenomes, there's epigenomics. There's so many different sorts. And these data themselves can be nested within other data sets. They're complex, they're variable, and they're scattered around the internet or in private repositories or public repositories in ways that it may not be immediately obvious for any particular researcher to find and locate all of the data that they may be looking for. One thing that's important around that also is that the databases themselves may not actually be connected and talking to one another. And that's an important aspect that our project is hoping to deal with. In order to understand this a little bit better, we have a scientific business analyst on our team, Kiva, and I'm going to ask her to take over sharing the screen, and she can explain to you the fantastic work that she's done in going out and talking to the community. And I will stop sharing so that she may.
Thanks, Catherine. Yeah, so I'll just go ahead and share. There we go. I think that's working. So yeah, as Catherine was saying, we went out and we spoke with members of the scientific community to try and understand how they access and use genomic data and what challenges they were facing during these processes. So we spoke with people across the scientific community because we wanted to build ARGA to be as useful as it can be to as many people as possible. The largest demographic we spoke to were life science researchers, so that's this chunk of the pie chart in blue, but we also spoke with people involved in industry and applications, so that includes things like biosecurity diagnosticians, as well as people involved in policy and regulation, as well as people involved in multiple of those sectors simultaneously. So the people that we spoke with also work in a wide range of different kind of scientific research areas. So we can see here that the kind of most prevalent scientific area or research area that people were working in was this biodiversity related research. We also spoke with quite a lot of people involved in collections and collections management. And as we were speaking to researchers and the scientific community, we were keeping a tally of the challenges that people were describing. And we grouped these into three kind of core categories with three kind of proposed solutions, as you can see here. So the first of these challenges being that genomic data availability is increasing exponentially. So as Catherine mentioned, there's lots of genomic data out there. It's being generated more and more on a daily basis. And we need searching and filtering capability to better be able to search through it and find the data that people are interested in. Secondly, data is stored in disparate and disconnected repositories. So individuals have to go to multiple online sites, go through them individually to find and access the data that they're looking for. And this is an issue that could be addressed with centralization of export and download capability. And thirdly, metadata can be insufficient and unstandardized. And here I'm saying metadata being information that's related to the genomic data. So that includes things like the occurrence location data of where, say, a specimen or an organism is from originally, or information relating to how the genomic sequence was generated. So, for example, the sequencing platform that was used. And one way of addressing this is curation of context and linkages to better give context about where that genomic data came from and how it was generated. And a bit more on that theme of context and linkages. It also relates to the challenges that researchers have described when speaking about trusting and reusing the data that they do find on the internet. So we've heard that researchers are faced with taxonomic uncertainty, which is where they can't be sure that the species identified is in fact the correct species and not a related species. Secondly, insufficient metadata, like I mentioned before. So that's where there isn't enough information about where data came from or how it was generated for a researcher to be able to reuse it. And thirdly is low or uncertain data quality. And that links to the above point of insufficient metadata, where there's not enough information available about how a sequence was generated in order for a researcher to judge the quality of that data. And so these three kind of issues clarified a bit of a secondary aim for ARGA, being to enrich genomic data with contextual information and resources to help users make their own quality assessments about how or whether they want to use a genomic data for a secondary analysis. So on the whole, what ARGA aims to do is to make data findable, accessible, interoperable, and reusable. So these are the FAIR principles, F-A-I-R, and they kind of underpin a lot of open science and open data, which helps researchers build on kind of pre-existing research to extend their own kind of findings. In terms of our vision for ARGA, what we want it to do is enable users to search for data from a range of existing repositories, but to do so from a single online portal. We want users to be able to explore the data according to metadata fields and curated groupings, and that relates a bit to the context and linkages that I was mentioning before. And thirdly, we want users to be able to access the data either by directly downloading it from ARGA to their computer or to export it to an online analysis platform. And in terms of that, I guess those three kind of big solutions that we mentioned before, so searching and filtering being the first one, what we want ARGA to do is access public data repositories, index the genomic sequence data in those repositories, process the accompanying metadata so that they're all in one standard or standardized to a single kind of formatting, and then represent that data on ARGA so that researchers can go to ARGA and then use it like a kind of search engine. They search for a particular thing, and then they can see what data relevant to that search is listed. And secondly, on that theme of context and linkages, we want that to underlie quite a lot of the tools and capacities that we're building into ARGA. So for example, taxon-based searching, which is where a user can search based on the organism name. Secondly, map-based searching, which is a bit like what's in the Atlas of Living Australia at the moment, where a user can search for something based on the location of that original specimen. Searching using other metadata features, searching by species list. So for example, the IUCN endangered species list or invasive species list, which are published by, say, federal government and state and territory government. And things that we're currently working on are ecological groupings and trait-based groupings. We also want to provide that export and download capacity. So we want to provide download capacity directly from ARGA. We want to provide export capacity to online bioinformatic analysis platforms such as Galaxy Australia, which are elsewhere on the internet. And where we can't provide direct download capacity from ARGA, we want to provide links to sources which can facilitate download from that direct source. And this is the case for things like generalist repositories, which take data that aren't just genomic data, because they're a bit harder for us to index. And finally, just to give you a bit of an idea about where we're at and where we're heading. So we're currently designing and building the front end of our portal. And just to give you a bit of a sneak peek, so this is a screenshot of our homepage as it is at the moment. So this is the search bar, and this is some of those kind of curated groupings that I was mentioning a bit earlier. And in terms of, I guess, our trajectory, we're currently in this kind of design and build phase, but we're planning an early release in July this year and hoping for a full release aiming towards the middle of next year. And finally, I want to acknowledge our kind of development team. So this includes Nick, our lead systems engineer, Chris, our data engineer, Goran, our software engineer, Matt for system support, and Mock, our UX UI designer, because they've been really busy actually building the Arga platform. And of course, Catherine, our project manager, who was speaking just before. And with that, that's me. So thanks very much, and happy to take any questions.
Lovely. Thank you, Kiva and Catherine, for that overview. That's really fantastic. So we now have 15 minutes or so, which is fantastic. Often people have so much to say that we run out of time at this section of the event. And people have been queuing up questions. I probably won't get to all of them, but I'm going to try and do them justice. I'd often try and group a few in here. I guess, and I'm going to put this to the panel. So whoever wants to speak first, feel free to jump in. So when my role is at the science team at the Atlas of Living Australia, and what we have a record of everyone who's cited the ALA from the scientific community, and what you notice in hindsight after 12 years of operating the system is that what the ALA enabled was people to understand where species live, and therefore to do things like project what had the impacts of things like climate change. And I wonder if you guys could give me a perspective on, so Erin was talking about epigenetics, Jess was integrating, 4 different kinds of data there? I mean, there's a lot of new information, and of course Arga's integrating it, which should supercharge that sort of approach. Does anyone have a feel or a view on the sort of insights that we'll be able to achieve in future from the sort of data you're generating now?
I can say something, Martin, if you like.
Sounds great.
Well, Arga takes all of the individual studies, like Jessica's in particular, is such a key example. So Kiva mentioned that there was a lot of confusion around taxonomic uncertainty. And what Jessica's actually done in her study is she's refined that taxonomy there at some point. So what we anticipate is that across the whole spectrum of life, from plants and algae and bacteria and higher vertebrates and everything in between, that we can slowly begin to refine these things. And that can only come through exposing what's actually out there. So in getting more access to more people to come and work with this, which will highlight any points where information is missing or it can be refined, we can actually begin to improve this process a great deal and offer the actual researcher a lot more certainty. The other thing with Arga that we're hoping is that as environmental DNA surveys become more commonplace and actually become de rigueur in the community for exploring an ecosystem, that we're able to give a lot more certainty around the sorts of individuals, animals that are existing, or plants, as it may be, in a particular time and space. We can then track over time how that changes, but that can only be done as we leverage the great wealth of stuff that's been built up in collections to this point in time, and then continue to invest in the collections so that they're there to be doing this work and bringing it in the one space. So my hope and my ambition for genomics is that it actually vastly increases and exponentially increases our capacity to understand how animals and plants have changed over time and space, and hopefully work with that to conserve them into the future.
Jessica, I could follow up on something that Catherine raised there. So she mentioned the example that you gave of, and you showed an example where a species had been split up and one where another 2 had been grouped together. I love examples like that. How prevalent is that? Should we expect a full turnover Australian taxonomy in the next 10 years? Is it that scale of revolution, or is this just tinkering at the edges to get a more clear picture of what our biodiversity is?
I would expect so, especially with groups that we didn't have a lot of understanding before, especially invertebrates, I would say. And with the genomics data, you're refining it. And when you do an integrative approach, which I think is the most important thing with the genetic data, and conciliate that with the other data that you have on distribution and morphology, you're actually able to get into these small pieces, put them together, and have a better idea of the diversity that you actually have.
No, thanks. I was fascinated by that. I guess going back to my previous question a little bit, Erin, you mentioned the epigenetic component and the ability to track the impact of environmental change, and now you're looking at it in historic samples. Does that mean that you're tracking the impact of past environmental change on those individuals? And what sort of insights do you think that will deliver?
Absolutely. It's our hope that we'll be able to utilize collections to essentially do retrospective studies of how species have responded to previous threats. And this will allow us to better understand how species cope and even start to generate biomarkers of species that are in trouble versus species that might survive a given threat. And this will help with management prioritization. If you know already that species have seen this kind of threat before, they've done okay, the biomarkers are indicating that they're doing all right, let's funnel money over this way to this species that is showing us indications that they're not going to be able to survive that next threat. And so we're hoping that it'll give that additional contextual information and not have to put feet on the ground as much, which at the end of the day is ultimately going to be the prohibitive factor.
No, it's fascinating stuff. If you don't mind, a couple of people have asked us, given the talk you gave about formalin and preserving an alcohol, have the work you've done changed the advice that you give or the practice you have for preserving things now? Do you still use ethanol and formalin, or is that still a recommendation? Has that become more useful, or is it that you now suggest something different?
So it has changed the way that we're starting to think about collections. So there's been a bit of a move away from formalin, because we know that it might be prohibitive for genomic sequencing in cases where we wouldn't necessarily take a fresh specimen. You wanted to avoid formalin, but now we're showing that not only can we get around it, but it may give you additional information than only having preserved it in ethanol. I would say don't shy away from formalin. Of course, take fresh tissue samples, lock them away in a freezer, use them for whatever you want, but so long as the specimen makes it into formalin fairly quickly, they're going to be useful for all sorts of downstream applications.
It's fantastic to know. And actually, look, I don't know if anyone else has memories of visiting museums as a child and seeing those banks of specimens preserved in formalin. It's slightly intimidating sort of a moment. It's fascinating that now it's become accessible in a new way through that technology.
It's a little macabre, so maybe more so than some of the other vaults, but I find it's one that is showing all sorts of opportunities because of that tissue specific information.
So going back to Catherine and Kiva, does the insights that Erin and Jessica and others are generating, there's a lot of new data coming online, and as you mentioned, and Kiva mentioned, in fact, that there's an exponential increase in data that you guys are seeing. Is presumably that has great richness and opportunity, but also presumably it's intimidating and difficult to handle that kind of data. Is there are you facing challenges in just storing that amount of information?
Well, we don't store, we're an index, so that's our immediate get out of jail free card there, is that we're an index, not a repository. But one of the things that we hope to be able to do is to standardize the format in which we are, one, displaying these data and interacting with them to be able to index them together. And we're using Darwin Core, and we're using a Darwin Core along an event-based model a lot more, so that we're able to reflect a little bit more of the nestedness in the nature of the extraction process and the collection process itself. So with Erin's examples of the tissues in museums, they've been collected at one point in time, and so there's a lot of metadata around that. Then there's the actual processes that they go through, and they can go through a number of processes, and different parts of that organism can be stored in different ways. So if we can create models that are extensible and able to flex to harness all of this information, we can then enrich it in a better way so that people can interact more accurately with those data and establish a set of key priorities for what constitutes the minimum data that you would want for a sample, and how to actually structure that in a way that it can then be reused by other scientists. Because one of the most disappointing and sad things that we find is I'm helping out the team by doing some work on looking through Dryad manually, and we've got an intern who's helping to do that with Zenodo. And we come across all of these data sets of SNPs. And there are as many different ways of making that CSV or Excel file as there are experiments to create the SNPs. And we hope to create a little bit of a groundswell around at least standardizing these formats so that, one, it helps researchers to structure their work more easily, and also make it available and reusable, most importantly, because it would be a very sad thing if people spend so much time and effort making these data sets, but then they can't be integrated into these meta-analyses later on.
Yeah, no, it's I've seen that happen in several different contexts throughout my career. Jessica, I wonder if that resonates with you at all. I mean, you described being in the field and using data from fossil specimens and being in the lab and in the museum, and you work for a museum. So you're sort of end to end in that sense, right? Are you seeing we talked about increasing technology for getting molecular information out and for storing that. Are you seeing comparable improvements in technology for identifying things in the field, storing data in standard ways, that sort of thing?
Yeah, I would say so. So in terms of storage, I think that is a massive problem that will come, that is already a big problem and it will continue to be. So if you imagine about the CT scans that I mentioned, for example, each image is really, really heavy. So you will need proper computers to store this data properly, and mitochondrial genomes or entire genomes as well. So it's massive. And in terms of looking for things in the field and try to relate to the taxonomy recent, so most of the things that we are describing now, especially related to reptiles, they are what we call cryptic species. So basically they're species that they look really similar if you just look at the body shapes, at the morphology in general, while if you look at the genetic, you see how distinguished they are. So these make things a bit more complicated. But as I mentioned before, if you use integrative approaches, then you can actually see what is this species from different angles. This also complements one thing and another. So it's the way I think to handle with this.
No worries, thank you. Well, look, that covers all the questions that we've had from the audience that I've seen. If anyone who's watching at home today has any further questions, I'm sure that the panel would love to hear from you and love to hear your feedback on the talks. Otherwise, thank you again to all our speakers. It's been fantastic speaking to you and hearing about your work. And join us again in 3 months' time when we run the next quarterly ALA webinar. Thank you so much. Have a good day.
Thanks for having us.
Thank you.
Thank you.